Good afternoon, guys. Today our topic is on glycolysis. In the previous lecture, we have discussed about the difference between hexokinase and glucokinase. I hope before watching this video, you should watch that video so that things should be more clear. And today we will discuss about the further important, most important step of glycolysis, which is conversion of fructose 6-phosphate to fructose 1,6-bisphosphate. So let's revise a little bit and then we can continue. First of all, glucose when enter into the cell is trapped in the form of glucose 6-phosphate. Okay, and this step occurs under the influence of two enzymes, either the hexokinase, which is present in most of the body tissues, or glucokinase, which is present in the liver. Hexokinase is regulated by the glucose 6-phosphate itself, and the glucokinase is regulated by the fructose 6-phosphate. Okay. The conversion of glucose 6-phosphate to its isomer, which is fructose 6-phosphate, is by an isomerase enzyme known as phosphoglucose isomerase. Further, the conversion of fructose 6-phosphate to fructose 1,6-bisphosphate is by the action of very important enzyme which is known as phosphofructokinase 1, and in this lecture we will abbreviate it as PFK1. Okay, phosphofructokinase 1, PFK1. This is the step of glycolysis. Okay, it's glycolysis proceeding forward. But this action can also be reversed despite it's a single or irreversible step. But in case of gluconeogenesis, it means that we are producing glucose from non-carbohydrate sources. It means that fructose 1,6-bisphosphate can be converted back into fructose 6-phosphate by the action of another enzyme which is known as fructose 1,6-bisphosphatase. I need you to understand that there are two kind of enzymes. First is kinase and other is phosphatase. What is difference between these two enzymes? The difference is that kinase adds phosphate group to the substrate and the phosphatase removes the phosphate group from a compound. Okay. It means that PFK1, which is a kinase, it will add a phosphate group to fructose 6-phosphate. And fructose 1,6-bisphosphatase, it means that it is going to remove a phosphate group from fructose 1,6-bisphosphate and it's going to convert back into fructose 6-phosphate. Okay. And these two steps do not occur simultaneously. It's very important to understand that when glycolysis is proceeding, gluconeogenesis cannot work simultaneously. The cells are not mad. They convert substrate to product and then they again convert the product back to substrate. It's not going to happen physiologically, right? Okay. I think logically that when the energy level of a cell is low, it means that the cell is not producing enough ATP. The cell is low in energy. It means that we need glycolysis. In these circumstances, we need glycolysis to produce a large amount of ATPs. When energy level is low, PFK1 is going to work, right? It will proceed the glycolysis downwards. But when energy level is high, it means that cell already has enough ATP to function, to carry out its normal activities, right? So we do not need glycolysis at that point, and we may need gluconeogenesis. We can produce glucose, okay? And in this condition, fructose 1,6-bisphosphatase can work, okay, and PFK1 will not work in case of high energy levels inside the cell. What is difference between bisphosphate and biphosphate? It's written fructose 1,6-bisphosphatase. It means that on carbon number 1 and carbon number 6 phosphate groups are present. but 1 and 6 are far apart. These are far apart. It means that bisphosphate means that phosphate groups are present on carbon atoms that are 4, and biphosphate means that the phosphate groups are present on the same number of carbon atom. Okay. The step we are discussing here, fructose 6-phosphate conversion to fructose 1,6-bisphosphate, is the most regulated step of glycolysis, and it's irreversible. As I have told you that it's an irreversible step. Either the step is going to function in this direction or it's going to function in that direction. These two steps cannot occur simultaneously inside a cell. So let's discuss about the allosteric regulation by PFK1, phosphofructokinase 1. Okay, it's allosteric regulation. What does meaning of allosteric regulation? It means that some molecules, some substances will influence the enzyme, will affect the enzyme without binding to its active site. They are allosteric regulators of that enzyme. It means that they are bind to allosteric site. They are not binding to the active site, but they will regulate the activity of that enzyme. So let's discuss one by one. Fructose 6-phosphate is converted into fructose 1,6-bisphosphate by the enzyme phosphofructokinase 1. I am repeating the same thing again and again for you to remember, for you to memorize it. Okay, this is the fructose phosphofructokinase 1 PFK1 enzyme, and this enzyme has several pockets. As I have told you that it is regulated by molecules, so it must have some pockets. Okay, these are pockets of PFK1. Okay, these are pockets. First pocket where ATP is going to bind. ATP is going to bind to this pocket. When ATP level inside the cell is high, do we need glycolysis or do we not need glycolysis? Think logically. We already have ATP, so why do we need glycolysis in that case? We don't need glycolysis, right? So ATP is an inhibitor of PFK1. Citrate. What is citrate? Citrate is a metabolic product of citric acid cycle, right? Citrate. So it means that citric acid cycle is working and citrate is being produced, and citrate, after the citric acid cycle, is going to produce a lot of ATP. So it means that we do not need glycolysis. It's also an inhibitor. What is AMP? AMP is adenosine monophosphate. It is the breakdown product of ATP. It means that ATP is shortage, ATP is less, ATP is not enough for the cell to function its normal activities. AMP is more. AMP is more, and AMP is going to promote glycolysis. Why is that? Because we need energy in this condition. Okay, these three are being discussed here, that ATP and citrate, these are inhibitors of glycolysis, and AMP is a promoter of glycolysis. The most important regulator, allosteric regulator of PFK1 is a compound or a substance that is very important. It's known as fructose 2,6-bisphosphate. It's similar to fructose 1,6-bisphosphate, but in the position of carbon number 1, there is carbon number 2 on which the phosphate is attached, right? In the formation of fructose 1,6-bisphosphate, for example, there are 10 fructose 6-phosphate. 9 will convert into fructose 1,6-bisphosphate, okay, and 1 will convert into fructose 2,6-bisphosphate, and this fructose 2,6-bisphosphate will promote the PFK1 to proceed glycolysis and convert these remaining 9 immediately to fructose 1,6-bisphosphate. It's a very important regulation by which this enzyme is going to proceed glycolysis forward. And there is also another enzyme by which fructose 6-phosphate is converted into fructose 2,6 bisphosphate. And this enzyme is like an hermaphrodite. What is hermaphrodite? Hermaphrodite is an animal or species that have both male and female gender on the same animal, right? It means that this enzyme is one, but it has two domains. It has a kinase domain, and it has a phosphatase domain. What I've told you about kinase and phosphatase. Kinase is a domain of the enzyme that phosphorylates something, and phosphatase will remove the phosphate groups. Okay. Conversion of fructose 6-phosphate to fructose 2,6 bisphosphate will require a kinase activity. It means that phosphofructokinase 2 (PFK2) will proceed this reaction. But this enzyme, this domain, which is fructose 2,6 bisphosphatase, we are talking about phosphatase, it will convert it back to fructose 6-phosphate, and its level is going to reduce, and its regulation to PFK1 will also reduce. Is this step going to proceed glycolysis? No, it will not, because fructose fructose 2,6 bisphosphate is going to be low, and in this condition the regulation to this enzyme will stop and the glycolysis will not proceed in the forward direction. Let's read about it. First of all is the ATP. When there is sufficient energy, we don't need glycolysis, right? So it's an inhibitor. When there is enough citrate, it's a metabolite of citric acid cycle, so we do not need glycolysis because enough citrate is present. When there is AMP regulation, it means that AMP is breakdown of ATP, so it means that ATP level is low, somewhat inside the cell, so we need glycolysis. When there is increased fructose 2,6 bisphosphate, it means that fructose 6-phosphate is converted into fructose 2,6 bisphosphate by the activity of PFK2, which is an kinase enzyme. It means that when its regulation is going to occur, it's a powerful regulator and it will promote glycolysis. But when fructose 2,6 bisphosphate is decreased, it means that it is converted back into fructose 6-phosphate by the action of phosphatase enzyme. Okay, its regulation on PFK1 will stop and glycolysis will be slowed down. I hope you understand this mechanism. It's a very important step of glycolysis in which the allosteric regulation is occurring by the PFK1. The second thing is the regulation of glycolysis by glucose level. Okay, in the blood, the glucose level can be varied. It can be high, it can be low, it can be normal. So how these glucose level will going to influence the glycolysis, we are going to study now. It means that when the glucose level is low, do we need glycolysis in this condition? Glucose level is already low. It means that insulin will going to be low. Okay, there is no glucose, so there will be no insulin. And glucagon is an also hormone. It's a compensatory hormone for the insulin. Glucagon will increase automatically. It will inhibit glycolysis. Think logically. When glucose inside the blood is low, in these circumstances, do we need glycolysis? Do we need to trap that glucose and convert it into pyruvate and then to citrate? Do we need all of these steps? No, we do not. But when glucose level is high, insulin is also high and glucagon will be low. It means that abundant amount of glucose is present in the bloodstream and we can convert it into ATP, so it will stimulate glycolysis. First we will discuss about when glucose is very low inside the blood. insulin will be gone, okay, and glucagon will appear. Don't remember it like glucagon will be high when glucose is no. Just remember when glucose is gone, okay, glucagon, then glucagon will going to be there, okay. Glucagon is there, it will stimulate the cell via the G stimulatory G stimulatory proteins, okay, G stimulatory proteins. G stimulatory will activate adenylate cyclase. It will going to convert ATP into CAMP, and CAMP will produce protein kinase A. And what's protein kinase A will going to do? It's a kinase. And what I've told you about kinase, kinase is an enzyme that add the phosphate group to something. Okay, it's going to phosphorylate something. What it's going to phosphorylate? It's that hermaphrodite. It's the same hermaphrodite. Okay, it's the hermaphrodite that has two like genders on the same enzyme. It has a kinase domain and it has a phosphatase domain, right? But when protein kinase A act on this, it will phosphorylate it, okay, and its kinase domain will going to be shut down, and phosphatase domain will going to work. What is the action of phosphatase? Fructose 2,6-bisphosphatase. Let's come back. Phosphatase will convert fructose 2,6-bisphosphate back to fructose 6-phosphate, and when its level is going to reduce inside the cell, there is no regulation to PFK1 and glycolysis will not proceed. Okay, it will going to increase the activity of phosphatase and it's going to reduce the activity of kinase. Let's revise, like read this thing we have just studied. It means that glucagon is there. It will affect through G stimulatory signal. It will phosphorylate the enzyme. Which enzyme? That hermaphrodite that has two domains, that has kinase domain and that has phosphatase domain. When it's phosphorylated, the kinase activity will slow down and phosphatase domain will work. It means that phosphatase activity is going to rise. It will convert fructose 2,6-bisphosphate back to fructose 6-phosphate. Fructose 2,6-bisphosphate was a powerful regulator, right? And when its regulation is stopped and it's absent, its level is reduced, it will inhibit the glycolysis. Because on PFK1, phosphofructokinase 1, its activity was very influenced, right? But when this compound is not present, how PFK1 will going to work? It will not work and glycolysis will be inhibited automatically. When glucose level is high, it means that we need glycolysis and there will be insulin, right? Insulin is present, insulin will come and it will produce a cascade of events, cascade of reactions and signals through the enzymes known as phosphatase proteins. Okay, what is the action of phosphatase? Phosphatase action is to dephosphorylate something. It will remove the phosphate group from something. It will dephosphorylate this enzyme. What enzyme? This hermaphrodite. And when this enzyme is dephosphorylated, its kinase activity will work and its phosphatase activity will slow down. Okay. And when its kinase activity is working, it means that it will convert fructose 6-phosphate to fructose 2,6-bisphosphate, and fructose 2,6-bisphosphate will influence the PFK1 to proceed glycolysis in this step, and it will convert a lot of fructose 1,6-bisphosphate. Okay. I am astonished that nature has put very remarkable functions inside our cells, that how these enzymes are regulated and how they are functioning. What are the allosteric regulations of that enzyme? How glucose level inside the blood can promote or inhibit the glycolysis via the action of these hormones, glucagon and insulin. Okay, let's revise all of that. First of all, when glucose is low, when glucose is gone, it means that glucagon will be there. Glucagon will stimulate G-stimulatory, it will stimulate adenylate cyclase, it will convert ATP to CAMP, it will produce protein kinase A. Kinase will phosphorylate something. It will phosphorylate this enzyme. When this enzyme is phosphorylated, its kinase activity will stop and phosphatase activity will rise. When its phosphatase activity will rise, it will convert fructose 2,6-phosphate back to fructose 6-phosphate. When its level is low, fructose 2,6-bisphosphate is low, the regulation to PFK1 will reduce, okay, and glycolysis will not proceed in forward direction, okay. But when there is insulin, insulin will act via phosphatase proteins. Phosphatase proteins will remove these phosphate from this enzyme. When these phosphates are removed, its kinase activity will work and phosphatase domain will shut down. When its kinase activity is working, it means that fructose 6-phosphate will be converted into fructose 2,6-bisphosphate by the fructose phosphofructose kinase 2, and kinase activity is working right. When its level is raised, it will regulate the PFK1 to proceed glycolysis in forward direction. So we need glycolysis, right, when glucose is high, when insulin is there. These all steps are very important to remember in case of glycolysis. And this was for today. And in the next lecture, we'll discuss about the next steps of glycolysis. I hope you understand this. Just try to make a concept about these things. Don't try to just memorize these things. Just make a concept. If the insulin is high, glucose will be high and it will work through phosphatase proteins. When glucose is low, there will be glucagon. It will activate the kinase A and it will phosphorylate these enzymes. And through these reactions, the glycolysis is either inhibited or is stimulated. So this was for today, and I will see you in the next video with